video link. I'll try to remember. It doesn't really matter on that. I mean, it won't like not when you submit it.
I'm not.
All right, so I appreciate those of you that were able to make it out to the candidate today, and I know that most of you are seniors and don't have a big stake in this, but if you do have time next week, Tuesday, Thursday, to come out, it's really important for those candidates to interact with students and to get that sort of going on. And we really, truly value your input. So even though seniors, it may not impact you, you guys have been here the longest and sort of know the most about the department and might have the best impression about who would be best to have. And those of you that are juniors, this person will probably teach learning and adaptive with the lab next year. So that'll be another option in addition to this course in cognitive and the new social. So, so you may have a chance to interact with them or do pieces with them as well. So, yes?
Is lunch actually provided by like after the presentation?
Yes. In between the teaching presentation, the research presentation, we'll have food available.
Oh.
Yep. Okay. Great. So, a little bit about what's going on today. We have lecture on sort of the history of how you go about adding a new taste category to those existing 5, right? And so there are 2 points of today's lecture, and then, so 2 points. One, just basic content about understanding how free fatty acids are detected in the mouth and how they influence behavior. But second, sort of how science works, right? So a lot of, like I said, a lot of parts of sensation and perception we still don't understand yet. So how do you go about figuring that out? How do you get new science into a textbook? Like how does it become common knowledge sort of thing? And so that's sort of what this story is about. How do we add another taste category to those listed taste categories, right? After lecture today, we're going to have stats session on our lab yesterday. And so I'm going to ask you guys to do some independent work on that because I think that you're capable of that, but I'm also going to give you a big gift as well. So it's going to be a little of both. I'll give you some stuff for free, and I'm going to ask you to do some work on your own with help from the TAs and myself. So you guys should be at that level where you're able to sort of perform on your own, that sort of thing. Cool? And then on Thursday, we have our next exam. And then we start our last section. I mean, we're almost getting towards the end, right, with the auditory system and the vestibular system. So, all good?
You meant the exams this Thursday. You said next Thursday.
Yeah, this Thursday. Next class.
Okay.
Yeah. All right. Cool. So, one of the things that I think, yes.
Is this going to include material on this next exam, or is this more of like
Yes, this is on, there's material on this exam. This material will be on the exam Thursday, and there will be points for this. So this is content that you guys are covering as well. So Stuart Feinstein is an olfactory researcher. I've known him for 20 years. Even though I don't study olfaction, the chemical senses hold a joint research meeting every year called ACHEMS. And it's the Association for Chemoreception Sciences, so it's all of smell and taste and oral nasal organ, all those researchers together at one conference. And he's up at Columbia in New York. He's a great guy, and he wrote this book that I love. If you're looking for a book to read, both of these books are very interesting. They're easy reads, they're short, called Failure, Why Science is So Successful, was his first book. And he basically talks about, people think that science is about you form a hypothesis and then you design an experiment and you test the hypothesis. That's not really how science really works, right? And he said it's kind of like, this quote is, being in a black room with no light looking for a black cat that may or may not be in the room. That's kind of what science is like. You're kind of poking around saying, this is what we know, and based on that, this is what we think might happen, so how can we see if we can find that stuff, right? And you're testing a whole bunch of things, and most of the time, it's failure. Most of the time, you don't find the cat. Every once in a while, you do find the cat, and then that's really important information, right? And so he wrote a second book that I think is equally interesting called Ignorance, which talks about the fact that ignorance, not being willfully ignorant, but not knowing things, is what drives science. And it's that quest to try and identify the things that we're ignorant about that's really important. And he sort of talks about all the different ways in which that drives different aspects of science. So today I'm going to show you some of the failures in our lab and some of the successes, but you have to break some eggs to make a cake, so to speak. And a lot of times when people come into thesis, they're like, oh yeah, we just like do this and it'll work, and it doesn't always work. And that's okay. That's all right. That's part of science, right? All right, so we talked about this, what's driving obesity, and there are three things, right? And we know that eating the French fries is bad for us, but why do we continue to eat them? And if you ask most people, they say because they taste good, right? Why do you eat high-fat foods that you know is not the best thing to eat? Because I like it, because it tastes good. So this sort of led us to this question, well, what makes it taste good? And historically, people have talked about fats as being rewarding in the gut, so coming through those, through the splanchnic nerve to provide calories, right? And then this triggers your reward system, and your brain then forms that conditioned preference that we talked about last time with the Kool-Aid and the sucrose infusions, right? So you eat French fries and they reward you from the gut, therefore it makes them taste better. But I guarantee I can give you something that is rich in fat that you've never tasted before. So you have no reward built up for that, and it will still be highly palatable, right? So it's not like that learning has to occur. And most of what people have talked about, the sensory aspects of fat, were that it was mouthfeel, that somehow it interacts with the touch system through this filiform papilla, and that it's not a taste, that it's not really going through the taste system, it's going through a different system. So our question was, can fat have a taste? And I mean, how many of you, if I brought in two little cups of whipped cream, and one was fat-free and one was regular, think they could tell the difference between the two? You tasted fat-free whipped cream, regular whipped cream, do they taste the same? They don't, right? There's something that's lacking, right? And it's not that Kraft hasn't tried to make the fat-free stuff. I mean, fat-free cheese, does that taste the same as regular cheese? No, it doesn't, right? And so what's missing in that? The fat's missing. And we're not saying it's not as rewarding, we're saying it doesn't taste as good, right? It doesn't taste as good. So for a long time, we've known that in cooking, fat appears to enhance taste. And I mentioned, I think this before, that if I gave you a plate of sucrose and a thing of Crisco, you probably wouldn't eat either one separately. But if you mix them together, right, that's essentially what this is. It's sugar and fat, and frosting is yummy, right? You get a scoop of that without a cupcake. And so there's this sort of common experience that, yes, fat produces a taste, and that taste is good. Right? So if we want to understand this, there should be a transduction mechanism, right? If we're going to say it's a taste signal, chemicals in fat have to interact with the receptor. So the first step is to say what chemicals are in fat. that could interact with the receptor. And it turns out that fats are triglycerides. Right? When I started doing this research, I was like, oh, I have to learn some chemistry here. And they have these fatty acids, 3 of them, that's why it's a triglyceride, 3 fatty acids bound to this glycerol backbone, and that's what makes a lipid. That's what makes a fat molecule a lipid, right? And it turns out there are enzymes in rats' mouths, particularly, that we've demonstrated, that break those bonds and release those fatty acids, and we call those then free fatty acids. And this was a study by Kawai and Fushiki that showed that if they put this trioline, which is a triglyceride with 3 linoleic acid fatty acids on it, that within 1 second, your tongue could generate 1.5% free fatty acids, right? So very rapidly, we can produce a concentration of free fatty acids that equals about 53 millimolar. If we talk about concentrations of solutions, about 53 millimolar within 1 second. And that'll be important, that 53 millimolar, because we're going to look at what the threshold to detect this is. And guess what? It's way below 53 millimolar. So within 1 second, a rat's mouth can convert these lipids into chemicals that in a concentration sufficient enough to taste them. All right. So if we say, okay, we don't really eat trioline, what kinds of fats do we eat? And corn oil is a fat that both we cook with. It's also a fat that's commonly found in a lot of foods and used in making a lot of foods. And so we started to look at what are the fatty acids in the triglycerides found in corn oil. And it turns out that there's a bunch of them. They're all kind of mixed in there, and there's 3s. It's not always the same one, right? But half of the fatty acids in corn oil are this fatty acid called linoleic acid, and 1/3 of them are oleic acid. And so together, these 2 fatty acids make up over 80% of the fatty acids in corn oil. And then there's these other ones that are kind of minor. But we decided, hey, if we want to see if we can detect a threshold for taste, let's focus on these as candidate fatty acids. They're the most prevalent ones in corn oil. Let's look at those first. That's why we chose those 2, right? Linoleic and oleic acid. Linoleic being the stronger or the more prevalent of the 2. All right, so in the mid-90s, researcher Tim Gilbertson was trying to see if he could identify a transduction mechanism for fatty acids. And his research model was to take taste receptor cells out of a rat's tongue, put them in a culture dish, right? Keep them alive, stick an electrode into the receptor cell so you can measure depolarization, and then put fatty acids in the solution surrounding it and see, does the taste cell depolarize or not? This was his method for trying to see if there's a way for fatty acids to depolarize taste receptor cells. Right? And so he did that, and what he found is that when he put linoleic acid in there, it blocked the outgoing potassium channel. It blocked this potassium channel, called a delayed rectified potassium channel. It's not really that important. But by blocking that, potassium built up inside the taste cell and it depolarized. Right? And so this is a potential mechanism now. It was the first time it was ever identified that fatty acids could potentially be transduced by taste receptor cells. All right, so this is his experiment here. Here's the taste receptor cell. He puts this is arachidonic acid, one of the free fatty acids, and then you see the potassium current, this is the outward current, drops. We're blocking potassium from going out. It recovers. He tried another one. Oleic acid didn't seem to act on that. Oleic acid didn't block that channel. Here's linoleic acid, again, blocks it. There were some other ones like palmitic acid that don't have this effect. So it was a selective effect for certain types of fatty acids. And he also wanted to know, well, what if I depolarize the cell And he was doing it with an electrical stimulus. That's why it's like a block here, right? He was injecting a little current in there. But you can imagine if you had that sucrose being transduced by that cell, what happens if we add fat, like frosting, we add the lipid on top of that. And when he puts the fatty acid on top of it, it enhances that signal. It makes that signal bigger, right? It adds to the depolarization. So here's Tim. He's a big Broncos fan. We don't hold that against him. He's always wearing Broncos stuff. And he did a whole bunch of experiments and identified not only that potassium channel, but he looked at a bunch of G protein coupled receptors, metabotropic receptors, that in other tissue, like the pancreas or the heart or the immune system or the intestine, we know that these receptors bind to fatty acids as a signaling molecule. So he wanted to know, well, are any of these on the tongue, and are any of these doing the same thing as a transduction mechanism on the tongue? And so this is his experiment, putting these are three different taste buds, fungiform, folate, and circumvallate, looking at the expression, whether or not those G protein coupled receptors are there, and he found evidence that they were on the tongue. And this is sort of the takeaway slide here, is that the potassium channel, that's the DRK, is found on all parts of the rat tongue equally. However, these 7 transmembrane metabotropic receptors were found in different parts of the tongue. Like the GPR43 was found on the anterior part of the tongue, but the GPR40 and 41 were mostly in the circumvallate, on the back part of the tongue.
So do metabotropic receptors release calcium?
Yeah, so they have a second messenger signal that gets released that stimulates calcium release for nourishment or exercise transfer.
So metabotropic will always be calcium, ionotropic will always be potassium, or no?
No, yes, metabotropic will always lead to calcium influx or calcium release. Ionotropic, in this case, is blocking potassium, which makes it positive. You can also have like salt, where sodium comes in, that would be ionotropic. So you can have other ions than potassium, but it's ion flow that is what's causing depolarization. Okay. So, yes, long story short, he identified these various different mechanisms, and all of this results in calcium, which could cause depolarization. So he effectively showed that there were both ionotropic and metabotropic transduction mechanisms in taste receptor cells from rats' tongues that could turn the presence of fatty acids into neural signals going to the brain, which was a big deal back then. So, review points. What do you need to know about this? Slide 11, jot that down, right? You need to know that they act through multiple pathways, right? They inhibit outward potassium current, and they increase calcium through metabotropic receptors, right? There's another one, another transporter protein that I didn't really talk about. You don't need to know too much about that, but the CD36 is an additional way to either recruit it, kind of like an olfactory system where they're bringing molecules there, or it might actually be another transduction mechanism, right? And each of these has different patterns of expression on the rat tongue, but they're all found on the rat tongue. All right, so at this time, Tim had done all this stuff in the late 1990s, and in 2001, when I came to Wofford and was going to start my research lab, I was looking at, okay, well, what research question am I going to answer? I'm not going to do the same thing that my PhD mentor did at Florida State, because he's doing that work. You know, I want to start my own line of research, and this was something that really excited me. And so I said, you know, in our lab at Wofford, we can answer some simple but important questions. Does dietary fat provide a motivation to consume things? So if I knew I could measure rats licking to sweet, and then I could measure rats licking to sweet plus fatty acids. Does it change that licking behavior? Does it enhance it? Does it make them want more of that? Right? And so some of the questions we wanted to ask were, it's detectable through the taste system. Fatty acids affect consumption of other things, like I was just talking about. And then we were also interested in whether or not, and I'll get to this sort of at the end, whether or not there were sex differences between males and females, and if there were genetic differences between obese and lean animals, because there's some evidence that people have different preferences, and this could be driving some of that obesity that we saw. So we went, our first experiment ever at Wofford was a really simple one, conditioned taste aversions, right? It's simple, but it's powerful. And we took rats and we gave them a free fatty acid, we gave them linoleic acid, we injected them with saline, and then we saw, do they consume that the next day? And then we gave them linoleic acid, and we gave them lithium chloride to make them sick, and we said, do they avoid it the next day? If they can avoid it, they must be able to detect it. They must be able to taste it, right? And we should be able to change the concentration to show that there's a threshold where they can detect it, and then if we make it weak enough, they can't detect it anymore, right? And that was our sort of paradigm. And so we did a very simple 2-bottle test. Just have 2 bottles there and measure, do they prefer or avoid the linoleic acid? And these were our results, and we had some control results here. So I'll just look at this graph first. So this is linoleic acid. This is oleic acid. If we look at linoleic acid, our highest concentration that we measured was 88. And sure enough, these are some like control solutions. But if we measure their taste aversion looking at this graph, we see that they avoid it compared to 1. And if we make it 66, they can still detect it and avoid it. And if we make it 44 micromolar, they no longer can show an aversion. So we're saying that that's too weak, it can't activate the taste system, right? So here we're saying their threshold was somewhere between 44 and 66 micromolar. And what was the amount of fatty acids that the rat's tongue can make within 1 second? 53 what? Millimolar, right? So they could detect 53 millimolar, right? And here we're saying their threshold is, we'll just say 44 micromolar, right? So we're talking about, if we were to put that in millimolar, it would be, micromolar, it would be that, right? So this is what gets made in 1.5 seconds on the rat's tongue, and they only need somewhere above this to be able to detect it. So we felt really good. We're like, hey, rats don't need a lot of this to be able to detect it and avoid it. We did the same thing over here with oleic acid and found a similar result, right? All right. So we published this. You can tell how long ago it is because I didn't have kids, so I didn't have gray hair yet. But we published this back in the early 2000s. We had students that came down to ACHEMS and presented this data, and we got our first publication from the lab. From a simple 2-bottle premises, no sophisticated equipment. All you needed was some lithium chloride and some syringes, some chemicals, and 2 bottles. It's really cool. Then we were able to up our game a little bit because we got some cool equipment in the lab that allowed us to do what we call brief access tests. So if you look at this video over here, the shutter's going to open and the rat's going to start licking to one of these bottle spouts. And the computer will count how many licks it does. I think it's a 30-second trial maybe. It's licking there. Rats can lick about 6 times, 7 times a second, really fast, faster than you and I can count. So the computer counts that. We can present 16 different solutions and do very brief access testing and look at a whole bunch of different concentrations within one test session. So then the shutter closes and it moves to a new position and it sort of starts over. And so in doing that, we could now look at low concentrations of sucrose to high concentrations. So now we want to say, can rats, does putting the fatty acids with another tastant affect their consumption of that tastant, right? And Tim Gilbertson's data would say it should make it taste stronger, right? Because if you have it, the current at one level and you have the fatty acid, it got up to this level, it got more intense. So here we have sucrose and glucose, two sugars. Rats like them. If you make them real weak, they don't really care. It tastes like water. As it gets sweeter, their licking goes up, right? And now we're going to add linoleic acid to it. And when we added linoleic acid, look what happened. The ones that they didn't like as much, they now like more. They lick more. It looks like the density went up, like Tim Gilbertson would have predicted from his model. And here, for glucose, again, they didn't lick as much. With fatty acids, they lick more, right? Until they kind of get to this maximal level of licking. We did the same thing with oleic acid, and again, this is 88 micromolar. So we're sitting over here, we're testing now. We're testing now with 88 micromolar, sort of our standard test. Way below the threshold. We did that with oleic acid, and we see an effect, but what do you see compared to linoleic acid? Is it as big an effect? No, it's a weaker effect. And that's going to be something that we see throughout, is that these fatty acids don't work equally, right? Yeah?
Isn't oleic acid more like found in more natural things like foods, whereas like linoleic acid is more like canola oils and like the...
Yeah, so linoleic acid is the primary fatty acid in corn oils and in vegetable oils. And oleic acid is the primary fatty acid in olive oil, which is why it's called oleic, from olive. It's the primary one in that. And if you were to taste, next time you're cooking or you're at your parents' house and they have different oils, taste corn oil and taste olive oil and tell me they don't taste different. They do. There's a different taste associated with that. And it's because they have different types of fatty acids present in these oils, right? Now we're going to look at those were things rats like. What about things rats don't like? So we make the rats thirsty, so they want to lick. And they will lick to low concentrations of salt, but we can make it salty enough that they say, even though I'm thirsty, I don't want to lick that. Same thing with sour, same thing with bitter, right? What would you predict if we put fatty acids on here? What would Tim Gilbertson's model say? What should it do?
Shouldn't it enhance the effects of, as it starts to dip lower into the fatty acid enhancer even more like that?
Yeah, so if we're saying that it makes it more intense, right? We would expect this to shift down. Like this one that they weren't avoiding, they should maybe start avoiding. And that's exactly what we saw, right? So things became aversive sooner. It looked like it increased the perceived intensity of both things they like and things they don't like. All right. So again, another group of students, we got results. We published this study. It's the second study that we published in the lab. And so, excuse me, your second summary slide, slide 23, write that down. All right? Do fatty acids affect other tastings? Yes, they increase the licking to things that they like and decrease the licking to things they don't, both of which behaviors are what you would expect if it made the perceived intensity stronger. It made the intensity of that taste stronger. Right? And, linoleic acid produced a bigger effect than oleic acid. Right? It was a stronger effect. All right, so we can check that one off. We answered that question. The other question was, are fatty acids detectable through the taste system? We already showed with the taste aversion, right, that rats could detect and avoid it. Right? Rats could detect and avoid it. But some people would say, well, they could be detecting that because the water feels different. touch. It has an oily feel or whatever. Or somehow there's a signal from their gut that says that there were fatty acids there, and that's how they're detecting it's not through taste. We want to prove it was through taste. So how are we going to do that? We went back to our conditioned taste aversion model, right? But now what we're going to do is cut one of the taste nerves. So we could come in and eliminate the chordotympany nerve, so the rat no longer had any taste signals from the anterior part of the tongue. And if it's tasting fatty acids, that should do what to the taste aversion? If we eliminate some of the taste signals. Make it easier or harder to detect it. Make it harder to show a taste aversion, right? All right. So this was our control data. So again, sodium chloride, lithium chloride, and we're showing now, this was with not 2-bottle tests, but in that device with just 8-second trials. So literally the animal has 8 seconds to lick and decide, hey, I gotta stop licking or I keep licking. And it was able to stop licking with 176, 88, at 44 again, it doesn't stop licking. It's like it can't taste it, right? With oleic acid, what happened? Did we show taste aversion? No. So that was our first failure. We were like, what happened there? We showed it with 2 bottles, why not with this? We'll come back to that. So we thought, well, maybe 8 seconds. So think about this. If this is a weaker stimulus, and the rat starts licking, it may take longer than 8 seconds of it licking it to recognize, hey, I gotta stop licking. In which case, it couldn't show a taste aversion. So we said, let's make it 30 seconds. That gives it enough time to stop licking, right? And show us that it can taste it. And so we did that, and what do we see? No taste aversion, right? And so what happened to linoleic acid when we made it 30 seconds? Yeah, well, all of a sudden, if we make it longer, they can detect it at 44, right? It just, with a shorter trial, it looked like they couldn't detect it. We made the trial longer, it could. So that's important when we talk about threshold, just like we talked about with your thermal lab, is that, which, by the way, I have 3 more labs to grade, and I'm working really hard. I'll get this done, if not tonight, tomorrow morning, and get this back to you. The method matters, right? The type of testing method can change where you think threshold is. So if we had only done this, we might have said, hey, threshold's here. If we'd only done a 2-bottle test, we may have said threshold's the same place. We now do a more sensitive test, and we can say, no, threshold's actually lower than that. They can detect it before that. All right, so now we cut the chordotympany nerve. We can't do much over here because we got a bunch of failure on that side. But here, when we cut the chordotympany nerve, this is the aversion. So what happened? Did we impair their ability to show a taste aversion? Yeah, right? And what about down here? Did we impair their ability to show a taste aversion? Yeah. Now, could they still show an aversion? Yeah. So we only cut the anterior part of the tongue. They still had the posterior part of the tongue that could be functional and working, right? So we did, but we were able to show, hey, there's signals, there's something going through the taste nerve, the chordotympany, that allows them to avoid it. So we were actually proved that the taste system, proves not the right word, but strongly support that the taste system is involved in this, right? I would say prove, right? But they still, and that other areas like the posterior part of the tongue are also involved because they didn't eliminate it, they were still able to avoid it. All right, so this bothered us, or me in particular, because in theory, this shouldn't have happened. And I wanted to understand why it happened. I said, well, if this is I was still hung up that this is a weaker stimulus. And I thought, well, 30 seconds should make it easier. But maybe it's not that they're not able to detect it. Maybe it's such a weaker stimulus, they didn't form a taste aversion when we tried to condition the taste aversion. They didn't associate the weaker taste with getting sick. So we did 3 consecutive days of giving them oleic acid, make them sick. The next day, give them oleic acid, make them sick. 3 times. And I said, if this doesn't work, they can't taste it. And when we did that, this is what we found for linoleic acid. They're now avoiding it. We started going lower because we hadn't found the threshold. 3 days, they can avoid at 20, but not at 5 or 2. So now we're down to say that the threshold is somewhere around 5. Wow, 5 total. We're now down to 5, and look at how much is made in 1.5 seconds. And guess what happened when we did linoleic acid? We were finally able, with 3 days of pairings, to show that rats could detect and avoid it. And again, like the threshold's higher, right? 20 instead of 5. So it is weaker, right? And it took 3 days of pairing that for the rats to learn, hey, this is the thing, the taste that's making me sick, right? So in science, like most other things, it's important not to give up if at first you don't succeed, to think about, well, is this really the way it is, the science, or am I doing something not correct? Can I change my methodology and show what I'm trying to figure out? We then cut the choroid tympani nerve, and again, we eliminate that condition taste aversion there. So, again, we published these results, another group of students, so we're like chugging along, publishing, where people are reading our articles. Other labs are now starting to do this. When we started doing this, it was our lab, Tim Gilbertson's lab, and one lab in France were the only ones looking at free fatty acids and whether or not rats can detect these. And now, we publish all this stuff, there's about a dozen or so different labs in the world that have started to look at this and do similar types of experiments. All right, so slide 33. Can this be detected through the taste system? Yes. If you impair the taste system, you impair the ability to show taste aversion. Right? And again, linoleic acid appears more salient. It appears to be a stronger, more salient is what we would use in S&P, right? Salient stimulus than oleic acid. One thing I'll point out is that these were all senior thesis types of experiments, and those students were working really hard. And when we started, our first publication was in 2003. This is in 2007. We did all that work within 4 years, so lots going on there. So the work that you guys do in your thesis group is important, and it's leading to publications. Even though your particular project may be a small part of it, those were 3 or 4 theses all wrapped up into that one experiment, right? So we can check that off, right? Okay, so it took us about 5 years. We produced 3 publications. All that was great. We then started thinking about, we had this idea, we want to relate this to humans, right? And obesity, and why do we consume high-fat foods? And do people that are obese consume more? Do they have a stronger preference than people that are lean? And there was some evidence then that there were more obese women than men. So is there something about sex that might interact with this? So we partnered with Tim Gilbertson, because now he, in the same period of time, has developed a genetic model for obesity in rats and is looking at their taste receptor cells to see are the taste receptor cells from fat rats different from lean rats. And so this is his stuff. He gave us this slide. So these are lean rats, and these are the fat rats. These are called OM, Osborne Mendel. These are the S5B strain. And again, in science, we often think about competition. My lab's looking at this, your lab's looking at this, and I want to publish before you publish, and we're all competing. But often collaboration is the best way to go, because I can do stuff in my lab that Tim couldn't do, and he has stuff in his lab that I can't do. Let's work together and create a better story about how all this works, right? So he sent us these rats so that we could have them here at Wofford, ship them to us. And it turns out that these rats have dietary preferences. The lean rats prefer a high carbohydrate diet. They actually prefer a high fat diet the least, if you give them a choice between high protein, high carbohydrate, and high fat. The obese rats have the opposite. They really prefer the fat diet and really show a difference to the carbohydrate diet, and the protein's about the same for both. So now we thought we had a model. Well, why do these rats not prefer fat and these rats do? Maybe they taste fat in a different way. Maybe they taste fat in a different way. All right. So in addition to wanting to know about these are the fat rats, these are the lean rats, we also wanted to know about males and females. And so we conditioned the taste aversion. We used our normal paradigm that I've been talking about and measured their aversion here to linoleic acid here. And it looked like there was no difference between the sexes or between the groups. And again, so we're like, okay, is this failure? Like we thought we'd see this big difference in preference. And we thought, well, if they can both detect fat, we're not saying one can't detect fat, right? We make them sick, they're probably both going to have a pretty strong avoidance to that. But if we then continue testing them and have extinction, maybe if we make the aversion not as strong, we'll start to see differences in sensitivity. So we continued testing them. So we didn't make them sick again. We just gave them the same test for another day. And when we did another day, we did extinction. When we did a test another day, all of a sudden, the females are now avoiding more strongly, and the males are showing less of an aversion. And the same effect here in the lean rats, the females showing a stronger aversion, the males showing a weaker aversion. And these rats, in general, if we overlay it with this, are showing a stronger aversion that the obese are than the lean. And if we go to day 3, we still see some of those effects, but now we've caused such extinction that they're really not avoiding that much. So we decided to hone in on that day 2, because that day 2 really looks like where we might be able to see differences in subtle sensitivity between these. They can both detect it, but is one more sensitive to fat than the other. And so if we overlay just the taste aversion, you can see that here we have stronger aversions for the obese compared to the lean, and that the dark lines, the males, are always higher within the strain than the females are, right? So within the strains, females more sensitive to fat than men, than boys, males, than the male rats, and within the sexes, the genetically obese rats are more sensitive than the genetically lean rats, which sort of fit with our paradigm. We also did another experiment where we raised fat. We thought, okay, well, what else could influence this besides genetics? What about experience? So we raised animals on a high-fat diet or on a lean diet. So growing up, they got a high-fat diet or a lean diet. And these are the same strains. And it turns out that if you raise a lean animal on a high-fat diet, they actually become more sensitive to that fat in a conditioned taste aversion compared to if they were on a normal diet. So the amount of fat that you consume somehow influences your sensitivity, makes it greater to detecting those free fatty acids. Right? So they actually even showed more sensitivity than the OM rats did. The OM rats didn't really show an effect of being on the high-fat diet. So here we're showing an effect of genetics. Here we're showing an effect of environment to influence fat sensitivity, fat taste sensitivity. All right, so we published 2 papers out of that with Tim Gilbertson as co-authors on that. And again, we're sort of chugging away. All right, so again, slide 43 is your summary slide of what it is you need to know for your review points. These animals had similar detection capabilities, right? They could both taste fat, but when we got into that extinction paradigm, we could see subtle differences in their sensitivity to detecting those fats. All right, so about this time, all of our work had been in animals, and because none of us, Tim or I, ever did human research and didn't really have much interest in doing it. But this guy, Rick Mattis at Purdue, ran a human taste lab, and he got interested in this. And so he did a bunch of different tests with different types of fatty acids, and found that across a whole bunch of fatty acids, he could measure a threshold for humans to detect that. He was calculating that in percent volume weight. If we convert that to our concentrations of molarity, that comes out to about 1 millimolar. So in humans, it looks like 1,000 micromolar or 1 millimolar was the ability for us to detect that. Now, does that surprise you that it's that much higher than rats? No, right? Because animals, we know animals, particularly chemical senses, are much more sensitive to that because they don't have other sorts of protective mechanisms, right? He also did some really interesting human tests to prove that it was taste and not olfaction. Like, how do we know it's not that retronasal passageway and stuff like that? So here, he looked at the threshold for taste. He looked at the threshold for orthonasal. So he simply had them smell a boat, that either a whey boat that had the fatty acid or not, and was able to determine the threshold for orthonasal smell. He then had them put that whey boat, open their mouth and put the whey boat in the back of their mouth so it couldn't touch the tongue, but could go up the retronasal passageway, so he could measure a threshold just for retronasal. So this is really kind of cool psychophysical stuff with humans to sort of tease out different ways in which that we could detect fatty acids. But the bottom line is that humans were showing this, right? So we thought, this is awesome. This would be in all the textbooks. We've shown it in an animal model, all this clear stuff. Rick Mattis now showing in humans that they have a threshold to detect it. And we present all this stuff to the scientific community, and they're like, well, wait a minute. All that's fine and good, but rats have this lingual lipase that hydrolyzes that Kawai Fushiki showed, right? And they said, well, humans don't have that. They don't have that same lingual lipase. So humans can't break this stuff down into fatty acids. And you guys are presenting fatty acids to them, but they can't break it down, so this isn't alcohol, right? Okay, if they were drinking free fatty acids, maybe, but they're not, they're eating lipids, and they can't break it down in their mouth, right? And so we then said, I think we can come up with a method to show that humans can break this stuff down in their mouth. So reluctantly, we did some human research here. And I teamed up with the chemistry department upstairs, because I didn't know. I was like, is there a way if I put fat, lipids, corn oil in someone's mouth, can we measure chemically, through chemistry, the amount of fatty acids that were produced? We're like, yeah, we can do that. We can do an acid-base titration. I'm like, what? So I had to learn some chemistry. And we decided that we could do this, right? And what we were able to do is we had human subjects take corn oil, swish it around their mouth, and then spit it back out. And then we tested that corn oil for the amount of fatty acid, free fatty acids that were present compared to corn oil that hadn't been in their mouth, right? And we compared that also to mineral oil. Mineral oil is an oil that doesn't have free fatty acids. So it was sort of our control to say we shouldn't detect any fatty acids after it's been in the mouth. Maybe the fatty acids come from saliva and not from the fat, right? And so what we found is this is the amount of fatty acids in corn oil, amount of fatty acids in saliva. If you add them together, you get this. And after 1 minute of swishing corn oil in your mouth, we were able to measure this. So we were able to measure a difference, an increase in fatty acids after 1 minute of swishing this around the mouth. And so then we said, well, what if we change the time? So we had people come in and swish corn oil in their mouth for 30 seconds, 1 minute, 2 minutes, 5 minutes, 10 minutes, and then some lucky volunteers got to swish corn over their mouth for 20 minutes. And then spit it back out, which gets a little tiring. I did all of this. But what we found is that we can produce, and again, this is 2 millimolar, right? Within 30 seconds, we can make 2 millimolar, which is twice the threshold that Rick Mattis showed, right? And if you keep that in your mouth for 45 seconds, it jumps way up to 4, and after that, it doesn't really matter. So while your mom said chew your food, if you chew it for 30 to 45 seconds, you're probably not getting anything else out of it if you chew it for 5 minutes, right? Again, as I mentioned, so we did it with corn oil, we did it with olive oil, and we did it with mineral oil. And with mineral oil, we don't see any fatty acids produced because there aren't any in there. So this is saying that those fatty acids are coming from the oil, not coming from the human body cell. Right? All right. So we tried to publish this, and I thought it was good data. But it got rejected for publication, which is not necessarily a failure, right? But they wanted us to take the samples after they were spit out and run it through a GC mass spec to show what fatty acids were present. All we can tell with this technique is that there are fatty acids in there, but we don't know that it's linoleic or oleic or what the fatty acid is. You have to do a chemical analysis of that in order to see what the molecular weight is. And apparently that's pretty difficult. We tried some experiments in the chemistry department here, and running human spit and corn oil through that machine is not good for that machine. It's not the kind of sample that it's used to. So the jury's still out on this. We haven't ever published this. But I think we have pretty good evidence, right, that this is, that humans can do this stuff. It's not something unique to rats. All right. So here are our review points. I'm going to wrap up early, right? We're going to have lab. We can start. Rachel's not here, which is fine. Let me just tell you what we're doing for lab. And if you guys want to get started on it, you can get a jump start, maybe get out of here early. And then I'll go back over it. Does that sound good? All right. And while we're not giving extra credit to come to the candidate talks, if you will come, if some of you will come next Tuesday and Thursday, I promise I'll have shorter lectures next Tuesday and Thursday as well. So we'll get out of class earlier then. Okay? That'll be my gift to you, no extra credit. But I won't say 65 slides in your throat. Like, where was that generosity or the patience?
It's usually the exam.
Probably the second division exam, which I don't know why. I think that's easier than the first division exam.
So not this exam.
And usually people figured out what to do, what they're doing wrong right now. All right. So after I go over this, I will go get the SPSS cart, and bring it down here. But let me just go over what we're going to do today. You know what? I didn't put my gift to you on here. So let me run down to I'm going to run down to my office. Grab the SPSS card if you guys want to take a quick break. I'm going to post my gift to you on the middle site, and then we'll get going on that. Okay, so let's get started at 2 o'clock. Sound good?